Okay, so I think we'll go ahead and get started here. We've got quite a number of people who have joined us already, and people will be trickling in. Hi, I'm Chuck Sanders. I'm here at Vanderbilt University. I'm the president of the Protein Society, and we're really pleased to launch our current academic year webinar series with a special presentation by Jeff Kelly and David Baker on protein scientists doing drug discovery. And we're really, really grateful to them for taking their time and making the effort to lead this first webinar. I'd like to note that we'd love to have your ideas for other webinars. It's not a lot of work to organize one of these. You really just need 4 speakers who are willing to speak and a good topic. And Raluca, our executive director, is willing to do the rest in terms of organizing. So if you're interested, please just shoot me or Raluca or the society an email, and we'd love to get your webinar in the queue. Let me just make a couple other Protein Society announcements before we get started. So this year's Protein Society Annual Symposium will be in July in 2023, not this year, next year. And so we certainly hope that you can make it. Those of you who made it to the San Francisco meeting in July know that it was really an outstanding meeting. And so we're really excited to be meeting back in person again. And so we really look forward to seeing you in Boston, and hope you can make room for this in your schedules. So it's time for our annual awards process to begin again. And of course, the first step is to call for nominations for awards. A new policy this year is that we've decided that it's not just Protein Society members who are eligible to nominate people for Protein Society awards. It's anyone. So we hope that you'll think about submitting a nomination of a distinguished colleague doing research in protein science. And you can find the link to the nominations page at the Society homepage. So if you just Google the Protein Society, it'll come right up. Nominally, the deadline is October 1st, but it will, as usual, be extended considerably, but it is time to start working on that if you're going to nominate someone. And I do want to call attention to our newest award, which is the Marie Maynard Daly Award. We're very excited about this award, which really honors a pioneering scientist, Marie Maynard Daly. And it is devoted to the interface between health and protein science. Human health and protein science. And so it's especially appropriate to mention that at this particular webinar. And we really are extremely hopeful we'll get a wonderful set of nominees for this inaugural version of this important award. Also just want to plug our journal, Protein Science. John Curian is now well ensconced as our editor-in-chief, doing a wonderful job. The impact factor is now 6.99. And we hope that you'll consider submitting some of your best work to this journal. And finally, thanks to all of you for attending and participating. And I'm going to now turn it over to David Baker, who's going to lead off here. And again, thanks so much for participating, and enjoy the show.
Let's see, can you see my screen okay?
Yes.
Yeah, okay, I want to thank Chuck and everybody else for the invitation to speak, and be fun if we were all in person, but I guess this is the next best thing. So I interpreted the topic of the webinar a bit liberally. Things are either sort of related to proteins as drugs, or they should hopefully inform our inform that in the not too distant future. So I want to talk about de novo protein design, which of course is the problem of making proteins which are completely unrelated to those that have come down through biological evolution. And there are 2 approaches. For many years we've been developing a sort of physically based approach to protein design, where basically trying to go based on the principle that amino acid sequences fold to their lowest energy states. And so in that model, you calculate the interactions between all the atoms in the protein structure as accurately as you can, and you design. And if you want, so if you want to design a sequence which folds up to a given structure, you find if you're interested in a particular structure, you come up with, you have to search for a sequence whose lowest energy state is that structure. And again, that's done using the Rosetta software developed by my group and many wonderful colleagues around the world, which basically has algorithms for sampling through sequence space and sampling through structure space and calculating the energies of different sequences on different protein backbones. Now, as you're all aware, deep learning has had a big impact in the protein structure prediction area in the last couple of years. And I'll show you in the second part of my talk how we can now use these deep learning methods for de novo protein design. So the first part, all the examples I give you will be proteins designed using Rosetta, this Rosetta sort of physically based approach where we first decide on a protein structure or function we want, then we find a sequence whose lowest energy state is that structure has the predicted function. And then we make the synthetic genes encoding the proteins, make the proteins, and determine their structures and functions. So it's a very exciting time for de novo protein design for a variety of reasons. One is, and that's very relevant to the theme of this webinar, is we have our first a medicine that's actually approved for use in humans. So it's actually going into people, which is exciting. So it's a COVID vaccine, and it's kind of an interesting example of how basic research can lead pretty quickly to applications. So let's see, it was about 5 or 6 years ago that we published a paper showing that we could design proteins design two protein components that self-assembled into icosahedral structures. And we really had no clue at that point what they would be useful for. At least, we had speculations. And my colleague Neil King then took these icosahedra and put the coronavirus receptor binding domain on them, and that's shown on the left here. And that turned out to be an extremely potent coronavirus vaccine that can be manufactured much more cheaply and distributed much more widely than the current generation of mRNA vaccines, because these are really stable proteins. Anyway, so de novo protein design is starting to actually have an impact on medicine, which is great. So also on the coronavirus side and proteins as drug sides, we've been working on this on the general problem of designing proteins that bind to arbitrary protein structures targets. So given a protein structure, design a small protein which binds to it. This is work from Longjun Cao and Brian Coventry. And so basically they were developing these methods, and then the pandemic broke out, and they applied them to make binders to the coronavirus receptor binding domain, which is shown here. And what they came up with were these very small proteins. So this one's 55 amino acids that this is a structure from David Wiesler's lab. And you can see this is the spike protein, and here are 3 copies of their protein bound. Then turns out that the protein design, the computational model, is very close to the design model. They're superimposed here. So we've gotten actually quite good at designing proteins which bind to arbitrary sites on targets of interest, and generally the structures come out very close. So to make this protein is a very potent neutralizer of the virus. To make it even more potent, Kei Wu, working with a group of others and collaborators really around the US, including Andrew Hunt and Mike Jewett's lab and Brett Case and Mike Dime's lab, designed these tripodal structures, which have 3 copies of the binding domain bound to, and they're geometrically matched. This is a theme of what we're a lot of what we're doing now is trying to geometrically match small design binding domains to targets, whether they're cell surface receptors we're trying to bring together and pick their orientations, or in this case, trying to get super high avidity binding to the virus. And this is again a cryo-EM structure from David Wiesler's lab, and you can see this 3-prong thing binding to the virus and to the spike protein. And because of the 3-fold engagement, this binds with extraordinary avidity. And it's we had found that the single domain was very potent and very effective at protecting and from infection in animal models. what we found for this threefold valent one with the super high avidity is that's essentially it neutralizes every escape variant of the virus that that has been identified thus far. So this one's behind the vaccine. It's headed for clinical trials as a nasal spray because super stable protein, you can formulate it, and it's quite effective. All right, so now getting away from coronavirus, but staying on the theme of binding, I told you we've gotten quite good at the problem of designing binders to sort of static protein structures. And Danny Sato took on the challenge of designing binders to flexible peptides. And the basic assumption was that that if you had a sequence which had some tendency to form a beta strand, an extended beta structure, then you could design a protein for that structure. And that's exactly what Danny's shown here, where this is his guest peptide, and he designs a protein that's in see my silly glasses. That surrounds it and basically has a slot open for it to fit in. Now he's actually designed binders to many different peptides, and we were particularly interested in amyloid forming peptides because, after all, amyloid has beta forming propensity. And so Danny designed proteins to bind to A beta, these are data from Thomas Knowles' lab in Cambridge, where they have a very nice assay for A beta aggregation. It basically removed and the conditions where the peptide would bind, it would aggregate almost instantaneously. And when you add even like when you add just stoichiometric amounts, even twofold lower than stoichiometric, which we don't totally understand, you get complete suppression of aggregation. And actually, we've been in touch with Jeff's lab, Jeff and his group, about these. We made binders to transthyretin peptides as well. So I think this is a pretty general way of designing proteins which bind to flexible peptides, particularly those with some amyloid forming propensity. Now, this is a little bit off topic for the theme today, but I just want to show you that we can design proteins to bind to small molecules too. And so this is work from Nate Ennist, and it's a particularly interesting small molecule. It's actually a pair of small molecules, and this is the chlorophyll special pair from photosynthesis. And so Nate's developed a general way of positioning, of designing things that will hold two chlorophyll molecules in different relative orientations to each other. And so this is really kind of neat because photosystems are really complicated, huge proteins, and now we can sort of just isolate, the key parts of it and study their photophysics. And this is a comparison of the crystal structure to the design model. It's very close. So as I illustrated in that first slide with the vaccine, we've been doing a lot of work in designing self-assembling structures, and Chunji Wang and Nate basically took chlorophyll, Nate's chlorophyll binding dimer, and used it as the edge in these octahedral structures shown here. So there's another protein, a trimer, which sits on the corner of the face. So this is kind of a neat thing. On each one of these edges, there's two chlorophyll molecules. And so we're very excited about using this to this sort of idea for artificial light harvesting complexes. So another type of self-assembly, these are also cages, but the difference between them and what I've described so far is that we've included a naturally occurring protein, namely an antibody here. And so in these structures, we have one design component, for example, this green pentameric protein that will bind to the side of an antibody, and it basically will assemble any antibody you give it into an icosahedral nanoparticle. And so we have, here's an octahedral nanoparticle, and this is work from Robbie Devine, who was a grad student in the lab. So we found that these antibodies, when you take antibodies and you hold them in these different types of nanoparticles, you greatly potentiate whatever type of agonistic activity they had. So, for example, these are results with a death receptor antibody, and we get rather potent potent enhancement of cell killing when antibodies are bundled in this way. But what I wanted to talk about today is our progress in using these for delivery, which is of course going to be important for drug development. So here's the octahedral nanocage with the tetramer and the antibody. in this case, I'm just showing you an FC to make it easier to visualize. And so you can see that we've got the tetramer on the 4-fold and the antibody on the C2, and there's an empty 3-fold. So Aaron Yang designed trimeric plugs that would sit on the 3-fold, and those are shown. That's shown here, and then is now a cryo-EM structure of this 3-component cage. And here we have her trimer. This is the plug, and this is the tetramer, and this is where the antibody sits. Now, what's neat about it is that she's designed this plug to be very pH sensitive. So these are just different variants, and you can see that they're assembling right in the range of the endosomal pH. So now we had very preliminary results with incorporating guide RNAs into nanoparticles and getting prime editing to editing within cells. And now we're trying to optimize that. And the nice thing about this system is you can put any antibody you want, and you can assemble a cage around it to use it. And so the antibody can help with the targeting part. So a challenge that we've worked on for a long time, so everyone knows that proteins form crystals, but it's been very, very hard to actually design proteins that will assemble to form crystals. And Zhe Li and Shunji Wang and Una Natterman have now solved that problem. And so basically the strategy has been to take a 2-component nanocage and then design it into a lattice and design interfaces between those components. They've succeeded in creating 3 different protein lattices. And these form so you just mix the 2 protein components, then they assemble first into the cage, and then they assemble into the crystal. So we're very excited about this sort of now being able to engineer protein crystals in a systematic way. This is one of the things, of course, that was very notable about proteins from the beginning is how irregular they are. And Tim Huddy was really interested in the question is, did that have to be the case, or maybe just nature didn't care about regularity? And so he set out to design these whole families of perfectly kind of perfectly straight proteins using, he designed first these helical repeat proteins out of perfectly straight parametric helices, and then simply replicated them along the chain. And I'm just going to show you some of the things he's made. It's neat because you can simply extend or contract them by adding or deleting repeats. So here's a square, a small version of a square, and a large version. Here's the cryo-EM, and he can also make ladders out of them. And so here you see images of the ladders, and again, the rungs, the spacing of these rungs and the length of the rungs can both be modulated. And we can now make nanocages with extensible arms. So we can start with small ones, and then just by adding inserts, grow the material. So this I think will make protein design much more modular. A lot of the reasoning now, we don't actually have to use Rosetta. A lot of it you can do by inspection once you know what the properties of the parts are. So everything I've talked about so far, the protein part has been static. Florian and Phil have now developed an approach to systematically designing proteins which have more than one state. So this is the basic idea. If you have a protein that looks like this, and if you generate another conformation that looks like this, even if this is the most stable conformation in isolation, if you add the appropriate, not beer can, but ligand, then you can get the whole thing to switch state. And they've succeeded in doing this really quite beautifully. So state X is the state that was on the left without the peptide, without the ligand, which in this case is a peptide. And here's the peptide binding. So we know that these undergo a rapid pre-equilibrium. Then when the peptide's added, you favor the state Y. And so here's and Phil and Florian have made quite a few designs like this, but here's an example of a crystal structure of a state Y with the ligand peptide in the bound to the what we call the hinge. So we've been using a deer spectroscopy with Stefan Stoll's group, and it's been really nice for the system. So we put probes on the two ends here, and their distance changes when between the bound state and the open state. And so we can experimentally measure the distance between the spin labels, and we see a clear, in different systems, we see different things. In some of them, we see actually population of the two states in the absence of the peptide. So they're really metamorphic proteins. And then you add the peptide and it collapses to one state. In this case, we get conversion from this apo state pretty quantitatively to the to the closed state when we add the this is the state X and this is the state Y. When we add the peptide, you can see that the two dyes are closer together when we add the peptide. And so Arvind Pillai and Abbas Idris have used this now to make dynamic oligomers. And so the basic idea is if we have a system like this where there's a ligand that binds to a protein, then if we build structures, architectures out of these, then when we add the small molecule, we should be able to generate changes. And this is basically designed allostery, where we're coupling, we're binding at distant from a site is changing interactions at a protein interaction site. And so here are just a couple examples of, here's an example of a system that forms a C2, a dimer, in the presence of the ligand when it's in the state Y, but a trimer when it's present in the X state. And so you can see here in the SEC data that you see this conversion from one state from the trimer in the absence of peptide to the dimer in the presence of peptide. And here's a system which goes in the reverse way from a C2 to a C3 when the peptide's added. Okay, so that's sort of where we are with design with Rosetta. And now I'm going to briefly tell you about design with deep learning. And so everyone knows about AlphaFold. We've developed a similar network called Rosetta Fold, and I'm not going to go through the details here. The original version of Rosetta Fold was not as accurate as AlphaFold. We're just about to release a new version which has similar accuracy, and I think will give different results. So in some cases it'll be less accurate, in some places it might be more. So I think it'll be a useful tool. Frank has now we now just put on just recently put online a version of Rosetta Fold that works on nucleic acids, and it's really quite remarkable. If you have given protein and RNA or DNA, it will assemble a complex. So it also predicts RNA structure. So Rosetta Fold 2 works with nucleic acids as well as proteins. Now, this is a horribly busy slide, but just to say we've been using deep learning as well, not only for the sequence to structure problem, but also for the structure to sequence problem. And so this is now, given a protein structure, design a sequence which encodes it. And this is kind of this slide is a little bit, I have mixed feelings about. So this is Rosetta after many years of work. This is for the standard test for protein design, where you have native structures, crystal structures, and you run your program, find the lowest energy sequence for the structure, and then you compare it to the native structure. And what happens is in the core of a protein, you tend to get you're very good at recovering the native sequence, but as you go further and further towards the surface, it gets harder and harder. Well, it turns out that this network that I described that Justus Duperas, postdoc in the lab, developed, is across the board, does a better job at designing at recovering native sequence. More significantly, we've gone back and taken many previously failed designs made using Rosetta and run protein MPNN and made those synthetic genes encoding those proteins, and the designs, we can often get designs that totally failed, that didn't express, and now they're expressed, stable, and functional. So it works really well. We've made it freely available, and I encourage people to use it for protein sequence design. So what I want to show you some examples of what we've done in the protein design area. So this is work by Lucas and Basil, which was actually just published last week. So you can, I'm just going to kind of gloss over it here. But so here, what they wanted to know is, can we use these methods to just freely explore protein space? So what they did is they chose a protein length, and they chose an oligomeric state, say 3-fold, 5-fold, 30-fold, and they kept those fixed, but they specified nothing else. And then they generated random sequences and predicted their structures. And of course, those structures aren't predicted to have any structure at all. But then they optimized the sequence by Monte Carlo, favoring sequences that were more predicted to fold to into a well-defined structure that had cyclic symmetry with n-fold symmetry. So they did this separately for different values of n and different values of L. And what came out of this were these beautiful looking structures. And this was done, we've done this both with Rosetta fold and AlphaFold. They did this with AlphaFold in this case. When they actually made the proteins, it was a colossal failure. So these AlphaFold design, these sequences at AlphaFold predicted strongly to fold to a given structure, just totally didn't fold at all. Then they took the protein MPNN program I described and redesigned the same backbones. And then like about half of designs, at least half of the designs actually worked. They folded up into new structures. Rob Ragotte solved the structures of many of these. These are crystal structures comparing these hallucinated structure to the design to the crystal structure. And so we really put nothing in, and this is just basically the network. We call it hallucination because basically we're simply trying to identify sequences that the network thinks fold up to some structure. It's kind of like taking a network that's trained to generate images of cats, or trying to identify images of cats and turning it around to give it like random images, optimize them till the network thinks it looks like a cat. And as you know, sometimes you get things that look like cats, and sometimes you get things that are kind of crazy hallucinations. And in this case, we're getting things that look like they could be proteins, and the crystal structures show that they actually fold. Now, Alexis Courbet, working with 2 rotation students, use this approach sort of on steroid to make proteins with making the repeat units smaller and expanding the number of repeats. So this one has actually 33 repeats. So there are 33 copies of this module, and they're predicted to form this structure. And so these are cryo-EM structures of 3 of these. And so they're these really huge rings with 10 nanometer diameters, and in all cases, the sequences and structures are really quite divergent from those of naturally occurring proteins. These ones look a little bit like LRRs, but they're these perfectly closed rings, and they have all these interesting and unusual extensions. So we're using these. Alexis has also been working on designing molecular machines. That's his day job, and I didn't have a chance to talk about that, but we're using these kind of hallucinated structures as the components. These are in the next generation of machines. Okay, so we can also use this deep learning methods to design enzymes. And so this is so Andy and Chris Norn set out to design an enzyme which would carry out this reaction, this luciferase reaction, by stabilizing this anionic species. And basically what Chris did is use the deep learning hallucination approaches to design families of backbones which have binding pockets, substrate binding clefts, just perfectly situated for finding this the substrate. And Andy made these proteins, and in the past we've had really hard times getting high activities right from the start by de novo protein design. But with just a couple in this case, we got designs that had quite a lot of activity, with just a couple mutations up at 10⁶ per mole per second kcat over cam, which we were very happy about. And and what's really neat about this design is that because it has such a compact binding pocket, it has very, very high substrate specificity compared to naturally occurring luciferases, which have much more open pockets. Okay, so the very final thing I'd want to talk about briefly is how we can design sort of proteins that scaffold functional sites. And there are 2 approaches that we've used to do that. One is to specify a site and then use this hallucination approach to basically search for sequences which are predicted to fold up into structures that have that site, for example, an enzyme active site. And I'll just give you one example of this. So this is in the vaccine context, coming back to the beginning. So here we have epitopes that that are recognized by neutralizing antibodies from respiratory syncytial virus. And this is now using this hallucination approach to scaffold these epitopes and basically design proteins that fold up so that exactly these structures are recapitulated. And this is work by Ju Wang, and he's made the proteins and they actually bind the neutralizing antibodies. So we're working with Bruno Carrera's group now to see if these can be developed as actually vaccines. And the second approach is kind of like, when you type in a sentence, start typing a sentence into your favorite word processor, it will try and autocomplete. And so what we've done is now to train Rosetta Fold, and it can do that because as a general model of natural language. So what we've done is now to train Rosetta Fold, deleting parts of the sequence and structure of each protein in the training set, and trained it to essentially recover missing information, the missing sequence and the missing structure. And we call this approach in-painting. And after the sort of analogous approach in image reconstruction, it's basically like, you could think of it like autocomplete. You just give in a functional site, for example, a metal binding site in this case, and then you're just giving that information. You're asking Rosetta Fold now to complete both the sequence and structure. And it works really well, and it's super fast. It's like, it's almost as fast as having your sentence auto-completed when you're typing into a word processor. So we're using this now to make a really wide variety of functional proteins. Okay, so I'm a little worried. I went long. Sorry, I just wanted to show you different things you could do with protein design today. So Longjun and Brian and Keja did the work, and Ina did the work on the coronavirus binders, the peptide binding work. The amyloid peptide binding work was done by Danny Sato. Nate designed the light harvesting proteins. The antibody cage work was done by Aaron and Robbie. Andrew's done a lot of the cryo-EM I showed you. Zhe and Shunji designed, and Una designed the crystals. Tim and Yang designed those modular materials. Thorian, Phil, Arvind, and Abbas designed those multi-state proteins that could be toggled by a ligand. I didn't have time to tell you about the molecular machines. The protein hallucination work I showed you was that of Basil, Lucas, and Alexei. Protein MPNN was developed by Eustace Dupras. The luciferase was designed by Andy and Chris. And Zhu and Sydney and Doug and Sergey did the work on the hallucinating functional sites. And David and Joe did the work on the in-painting method. And Rosetta Fold is the work of Ming-Kyung Bak and Frank Mayo. I also want to acknowledge I've had a huge number of colleagues who've made important contributions at the Institute for Protein Design: Lance Stewart, Asim Bera, Lauren Carter, and others. So I hope I didn't go too long, but I'm happy to take any questions, or we can do that afterwards.
David, your timing was perfect as always. This is really a phenomenal accomplishment now that we have multiple approaches to design proteins quite efficiently. Congratulations. While the questions are being typed, I was wondering how you guys are thinking about sort of rationally perhaps using what we know about the immune system to avoid protein immunity, to start to think about using these algorithms to design things that won't be recognized by the immune system as foreign.
Yeah, that's a really, of course, critical question. And there are basically two approaches. One is what we call sort of implicit negative design. That is, design the proteins so that they will have physical properties which will make them less immunogenic. So that means very, very stable, small, highly, very polar, so less likely to be taken up into dendritic cells and less likely to be processed. And that's the approach that we've used for the most part. And that it seems to work. So we have a couple of proteins now in clinical trials, and I should say in animal models we haven't seen large immune responses. Sometimes we see very modest ones. And the proteins the couple designs are now in humans as therapeutics. So we'll learn a lot more. The second approach, of course, is to actively design out T cell epitopes, and things that are likely to be presented on MHC. So we've been, we and other people are working on deep learning approaches to predicting, peptides that peptide MHC engagement. And it's not clear at this point whether that will be necessary or not, but that would be obviously the way to go. The problem is the human population is so polymorphic. It's tough to do that. Much easier to do it in a in a mouse strain that just has one HLA allele.
Maybe Chuck, why don't you go? And then let me just say, before you ask your question, the attendees, please type your questions in the Q&A box, and I'll do my best to convert them to questions for David.
So David, I had a question about your vaccine development project. So obviously you guys designed the basic particles that would be used as vaccines. I'm just curious, how does it work? Do you hand that off then to the real experts in drug and antibody development, and you never hear from them again, or has it been highly interactive between your lab and the antibody developers all the way through the clinic, all the way to the clinic?
Yeah, well, we started this Institute for Protein Design in Seattle, and we hired Neil King, who's now my colleague, and his lab is really focusing on using protein design for vaccinology. So he has become, a total expert on all the steps needed to go through. So he's worked very, so basically he's now, both designing vaccines, he's doing the animal experiments. He's got a network of collaborators really around the world who are, we can do the simple animal experiments, but then, you go through higher animals, and then starting to learn more about the whole, clinical trial thing. So he's really become a world-class expert. So that's the answer.
Thank you.
So, David, the first question is from Diego Ferrero. Can you please comment about the actual folding process of the design proteins? Are they, quote, bad folders as top 7?
Yeah, that's a super interesting question. And I'm glad you brought that up. I think, there's always a question of what have you learned from de novo protein design? And I should say, both with the Rosetta approach and with the deep learning approaches I talked about at the end, we never have considered the actual dynamics of folding, the process of folding. We've always designed solely for the final folded state to be, either very low in energy in the case of Rosetta, or strongly predicted by the network to fold in the case of the deep learning approaches. So I think the fact that we so I think what this proves, and we've designed hundreds of proteins over the years, and the structure almost always comes out right. So I think when we have successful design. So what I think what that means is that thermodynamics kind of trumps kinetics in protein folding. So if there is a sufficiently low energy state, the sequence will get there. Now, in some cases, as in the top 7 case you point out, there are, it's not as cooperative as native protein structures, but, and it's really kind of dependent on the size of the protein. For the smaller proteins we're designing, they really seem perfectly to state. And Gabe Rocklin, who's now at Northwestern, has done really an extensive analysis of the landscapes of these designed proteins. So, but I think there is still more to do there. So we haven't investigated the folding of most design proteins as extensively as we did for top 7, but Gabe and his lab are really developing methods to probe that on a very large scale.
An anonymous question, very interesting research work. Could you talk a bit about the protein-protein interaction that is responsible for the protein binding? And also, where do you see the squares, rectangles, and other geometric shapes being applied?
Yeah, those are both good questions. Okay, so the basic principle for protein binding is you're given sort of a structure of an arbitrary protein of interest, and now someone points to a particular region on that structure and says, design me a protein which will bind there. So then basically the challenge is to design a sequence which will fold up into a structure which is shape complementary and chemically complementary to that structure, basically making very low energy interactions. And so in the Rosetta-based approach, what we do is we generate very large numbers of scaffolds on the computer, dock them against the target, then design the interface for maximum shape and chemical complementarity. And then we make the proteins and measure binding. Now, one of the important things I didn't mention to say is most functional designs actually don't work. So we have to, so protein design is is very far from perfect. So we generally have to test quite a few designs to find ones that work. And now with the deep learning methods, it's well, it's a little bit different, but the basic concept is the same. Now, what are the applications for these square and perfectly straight proteins? Well, if anyone has any good ideas, can you send me an email? Because we're trying to figure that out now. But we can imagine, like, one of the ideas, what if you had lanthanide binding sites in each repeat? Then you could make things that you might be super radiant or have other properties. So yeah, we're trying to figure out what the applications of a general protein design building kit is, like Legos.
Another anonymous question: how do you design algorithms for auto-completing unknown protein sequences around an ionic binding pocket?
Yeah, so what we do there is we start by specifying the residues and the geometry around the ion binding site. So now we have, we have part of the protein specified. And now in conventional protein design, what we always would have had to do is decide, well, what secondary structure elements were we going to use to connect them? But the model has now been trained to take bits of protein and just complete them into full structures. And so it's really the same thing as, you type the cat walked into your word processor. I'm surprised my cat isn't crawling all over me, which is climbing all over, which she threatened to do about 10 minutes ago. And then the network will complete it because there's a pretty good idea of what the high probability words are that you're going to type after that. In the same way, once you've built this metal binding site, the network has a pretty good idea of, from all its experiencing protein sequence and structures, what the rest of it's going to look like. And it really, so we don't know the network is this huge black box, but it's been trained to do this. So you put in a partial structure in, and out comes a complete structure and sequence. So that's what it's been trained to do. That's one of the things a little dissatisfying about deep learning is you can't tell exactly what it's doing, right? Or how it does it.
A question from Vera Rana. Great, amazing talk. My question pertains also to the autocomplete method. Is it possible to use the imprinting method to autocomplete the full range of structural motifs?
Yeah, it's really agnostic. You don't have to specify the topology in advance. It will figure it out for you. So depending on what the arrangement of what the fixed part looks like, it would complete it into an alpha helix or alpha helical structure, or beta sheet structure, or alpha beta structure. It's sort of like, again, it's got this partial structure, and it's kind of reasoning about, given this partial structure, what would be the most, if this were a native structure, would be the most probable way to complete it. So it really works over all motifs. So again, before we would have had to decide on the structural motif, but we don't have to do that anymore.
Anonymous, very interesting. Thank you. Could you tell us a bit more about the method process of designing functional enzymes?
Right. So in that case, you start off by, you choose the chemical reaction, you identify the transition state, and then you design a sort of an ideal active site. Working with Ken Hauken for many years, we call them theozymes. Can coin that term. And so now you have sort of an ideal active site that stabilizes the transition state for that reaction. What we used to do at that point was to search through protein scaffolds, like native protein scaffolds, trying to find a place where we could put it. But now we can use these deep learning methods to assemble like whole families of structures that have that active site. And we can do that using the several different approaches. But the in-painting approach, for example, you would specify the active site and just grow in the rest of the protein. And then in what I showed you for the luciferase, what we did is what Chris did was to generate whole families of structures that had a pocket that was roughly the right size and shape for harboring the luciferin substrate.
A question from Jermaine Davis. Do you think the two-component nanocages can be fused to proteins to aid in their crystallization?
Yeah. So the other thing is for cryo-EM, and Todd Yates had a very nice paper a few years ago, basically showing that you basically in the cryo-EM case, you can average the information on you can get the cage part, and then you can get a structure of what's stuck to it. We're really interested now in using those designed crystal lattices I showed you as hosts. So you put your unknown protein in as a guest by genetic fusion, and we're working on that now. We're trying to get the diffraction, the resolution up, but I think that would be another approach to try and solve structures of unknown proteins.
Anonymous, is there any concern that the proteins you design can be too stable in the sense that the cells cannot readily break them down?
Yeah, well, I think, they are proteins made out of the natural 20 amino acids. And I think, with things like the proteasome, energy dependent proteases, they will all be broken down. We have what happens with the small proteins is they get excreted through the kidney too, so the body eliminates them. So we haven't really seen signs of proteins lasting for a really long time in cases where we don't want that to happen. One place where that's more of an issue is I didn't talk about today, but we've been designing proteins and really more macrocycles using unnatural amino acids, and those are very stable structures. They're very rigid. That they're very interesting as they get across membranes, but they aren't going to fit into protease active sites as well. So degradation might be a problem, but on the other hand, your body again eliminates them like it would any small molecule. So,
Anonymous, mind-blowing amount of work. That's certainly the case. I was wondering how to go about the de novo design of enzymes through multiple conformational states.
Yeah, that's a really good question. So that's what makes designing enzymes harder than designing binders, because an enzyme, these are multiple states. There's the substrate, you have to bind the substrate, then selectively stabilize the transition state, then release the product. And that is and so enzymes, natural enzymes are essentially masters of the art of compromise. So that is, we're working on multi-state protein design. But the problem is there has to be dynamics, but it has to be the right dynamics. So you have to very selectively stabilize the transition state relative to the substrate. So the answer is we are developing methods to do it, but it is one of the real challenges for enzyme design, considering the whole reaction pathway.
So from Matt Sinclair, how well does the hallucination scaffold building handle trickier ligands or cofactors such as metal clusters?
Yeah, that's a really good question. So the way we're doing it now is for the metal cluster, we take, again, we position side chains around it, and then we basically try and design a protein which positions those side chains in the right place. But we really want, what we're working on, I think the longer term solution of this is to have Rosetta Fold understand small molecules and metal clusters and so forth. So it will be able to, you would be able to give it the metal cluster, and then it will sort of know, it'll know what sort of interactions to make with it. So I think we really would love to collaborate with people on this problem, because metal clusters, of course, there's also the issue about how just the assembly and incorporation into proteins. So we're trying to work on that now. Actually, it's going to be a rotation student project this quarter, and we'd love to collaborate if people have metal clusters that know how to work with them and so forth.
So Anonymous, David, how do you deal with intrinsically disordered regions when these intrinsically disordered regions are critical for function?
Yeah, so there some intrinsically disordered regions, will actually be ordered under if they're binding to a protein. And so I showed you with the example of the amyloid peptides that we can now design proteins that bind to peptides that would normally be disordered, and they become ordered upon binding. Keija Wu also has done very nice work I didn't have time to talk about in designing binders to quite polar stretches of peptide sequence. And so we're getting better at designing binders to flexible peptides. Now, we're of course binding them in an ordered conformation. There are other intrinsically disordered parts of proteins that don't have a well-defined structure in any state and are very difficult to stabilize. And those, I think, will be hard to bind. But I'm pretty optimistic that Keija's approach will kind of let us solve the sort of general peptide recognition problem.
Anonymous, it's absolutely amazing to see the possibilities. And the question goes on to talk about handling multiple conformational conformations, which of course are required for enzymes. And you've already answered such a question. The end of it is, how would directed evolution cycles weigh in enhancing performance of conformational switching?
Yeah, I think the way that directed evolution would play in is, say, with an enzyme, for example, Don Hilbert's group has done quite a lot of this and beautiful work, and Danny Toffick's group did as well, where if you take a design that has relatively low activity, you can apply directed evolution to optimizing the activity, and that will have to optimize the dynamics and the balance between the different states at the same time. Now, that's not the same as using directed evolution to explicitly favor a particular ratio or multiple landscape. And I think that's harder. It's easier to use directed evolution on functional readout.
From Matthias Buck, one criticism of machine learning is that it is a bit of a black box. What approaches are you taking to abstract/understand the rules of protein structures, protein-protein interactions?
Yeah, that's a really good point. And I think the physically based, the Rosetta approaches, exactly what you get. You can trace back to, these hydrogen bonds here, or, this steric, clash in this state favoring the other one. I think in the with the deep learning methods, what one can still do is sort of do mutational analysis. So you can figure out, what amino acids the network thinks are important for things. But really understanding fundamentally what's going on in the network is tough. But on the other hand, we think, well, okay, we have, physics and math and chemistry, and that's all these principles, and we can think about things that way. Well, our brains are our brains are neural networks, and it's really, really hard to understand how they work. So we're familiar with things that are very complicated and have a very good reasoning power, and we've been living with them for hundreds of millions of years. So yeah, but it doesn't have the transparency that physical models have.
From Francis Vallevetto, what advances in unnatural amino acid incorporation into proteins do you anticipate using unnatural amino acids in your design process to generate catalytic activities beyond the naturals?
Yeah, so right, so that's eminently doable. That's a really good case though. There's a difference between the energy-based and the deep learning-based approaches. So from the Rosetta point of view, it's really easy to extend unnatural amino acids because they're all made out of atoms, and you have models for how to compute hydrogen bonding energies and van der Waals interactions. And we've actually Jeremy Mills, who's now in Arizona, used unnatural amino acid to incorporation to design new functional proteins. And so we do use unnatural amino acids using genetic code, extension approaches developed by Pete Schultz and others. But where we've really been using them the most extensively are, as I mentioned earlier, are for systems that we can chemically synthesize. So peptides below 45 amino acids, because there you can include unnatural amino acids. You can even change the backbone, so you're not confined to a peptide backbone. And the basic principles that we use in Rosetta still hold. So Patrick Salveson has designed this whole class of what we call mini cycles. Like, they look like small molecule drugs, but they have the same kind of properties as the designed proteins. With deep learning, it's hard because there are fewer examples. So we're working to extend these deep learning models to general, non-protein chemistry. And there the training sets are much smaller, so it's harder.
From Manfred Phillip, your methods be used to predict the energetics of protein ligand interactions?
Let's see. Well, that's a really important question. I think that so there's the deep learning methods. You would need to, so the deep learning methods, what you would need for that, and so deep learning methods are only as good as their training data. So you would need a very, very large set of small molecules bound with really accurate affinity data. And as everyone knows, those sets are kind of variable. You would also need crystal structures. So that, I think, using deep learning methods is hard. And then with Rosetta, Rosetta is calculating energies, but not really free energies very well. The solvation part of is included, but not the conformational part. So, the most accurate method is probably still free energy perturbation calculations where you're actually modeling the you're getting entropies as well. I think Rosetta is reasonably good at calculating delta G's, but absolute free energies of binding, which you need to calculate affinities, is hard.
The next is from Shugong Zhang. Can you design small protein enzymes to do photosynthesis as in the origin of chemical life?
Yeah, we're working towards that. I showed you that we're trying to design these reaction centers, and say with the metal cluster thing, if you get the manganese water splitting complex, I believe that. I mean, I think there's a lot of steps there. So really, we're really just very, very, very much at the beginning. But I think it's a great challenge for protein design. There's so many interesting things that happen during photosynthesis, but one should not underestimate the complexity of the natural process and how sophisticated it is.
From Louise Garone, a follow-up to Chuck's question. What are you doing to prepare students for careers in the biopharmaceutical industry? Or do you anticipate they will all have careers in the academic side of partnerships working through world-class experts?
Yeah, I think increasingly the people coming to my group, students and postdocs, are interested in starting companies after they leave. Not everybody, but there's more interest in that than there was, say, 10 or 15 years ago when most people wanted to become professors. And I think there's a lot of, and I think what's happened is there's a lot of interest now in de novo protein design and the medical applications, and actually applications and materials as well. So people have been able to start companies, quite a few companies that are, exploring applications to cancer, to vaccinology, to autoimmunity. And then other people in the group who don't want to start companies frequently will just, go as employees to these companies. So we're kind of creating an ecosystem which is for answering exactly that question.
Anonymous, how do you think about designing membrane proteins, and how's it going?
Yeah, so we're making a lot of progress on that. So we've been looking both at helical transmembrane proteins and beta sheet transmembrane proteins, and like outer membrane beta barrels. And basically using the Rosetta approach, we just have to change the physical model of the part within the membrane. So we've been able to design alpha helical, de novo alpha helical protein channels that function in mammalian cells. And we've also been able to design beta barrels that have different pore sizes. So we now can design quite large beta barrels from scratch, which have very big pores. And we've recently figured out how to incorporate them into membranes. We get beautiful single channel conductances. And so we're now very excited about making new classes of pores for things like, DNA and protein sequencing and sensing a wide variety of molecules. And that's been a great collaboration with Anastasia Vorobyeva's group, who really cracked the problem when she was a postdoc in my lab and now has her own group in Belgium. And Samuel Levin and Sagardip and my group are continuing to work on that.
Ahmad Azit, how about designing new proteins that are metamorphic?
Yeah, well, I talked about that. We for these proteins have a closed state and an open state that I showed with the beer can. So we've designed some of those which very clearly populate both the open state and the closed state at equilibrium. And then you add the peptide, and they all open up to accommodate the peptide ligand. That was the other beer can. So and there what we do is we design the energetics of the system so that both states are populated. We can do that. We sort of do it from an energetic basis with Rosetta, or with the deep learning methods, we find sequences which actually are predicted to populate both states. And it's actually kind of neat. One way of testing that is we can design in disulfides, and then depending on whether the disulfide is present or not, the deep learning method will predict it to be closed or open. So I think that's really come a long ways.
So I'm going to have to be a little selective here, being mindful of time. So
we need time for your talk.
Great talk. I'm just wondering if you have any database where you dump the designer of the proteins that did not pan out. Is this a useful learning paradigm?
I think it is very useful. And our best example of that is for the protein-protein interaction work, where we, as part of the paper, we made available all the designs that we tested, and we tested very large numbers because this was sort of from the beginning when we weren't so good at it. And so we provide all the sequences, the design models, and the binding data we had. And so we do make those available, and I hope people are using them for I'm sure that we haven't, there's still a lot to figure out and learn about what's the difference between designs that work and designs that don't. And so I really encourage people who have an interest in this to look at that data. And certainly I'll be very, very interested in what people find out.
So Jonathan Sheehan, I saw an EF hand as one of your examples. Have you succeeded in designing major conformational changes like calmodulin upon ion binding?
Yeah, that's a really good question. We actually haven't. We're trying to do that now with those proteins that undergo conformational changes like this, to design small molecule binding sites. And in one of the approaches to this, we're collaborating with Tanya Kortema's lab at UCSF. So I think it's a really exciting area to sort of metal-induced conformational change. The things we've designed so far have been pretty static.
Patrick Ellen, how does deep learning handle the less common structure motifs such as the non-glycine amino acids in the disallowed regions of the Ramachandran plot? Yeah,
well, it's as people have noted in the questions, and as I mentioned, deep learning methods are only as good as their training data. So the answer is, if it's something that's really not seen in natural proteins at all, then the deep learning method will likely not get it right. But there's so many protein structures. I mean, the success of the deep learning structure prediction methods are really a testament to all the work done by structural biologists. I mean, even relatively rare, there may be 100 examples in the PDB, and that could be enough for the networks to get these right. And so, so I think what sort of the success of the structure prediction method suggests that in some sense, the PDB is complete in terms of providing a mapping from sequence to structure for things just made out of the natural 20 amino acids.
Iris Smith asks, from a macromolecular perspective, what are you currently doing to perhaps combine hybrid quantum mechanics, machine learning neural networks for large systems? For example,
that's a good question.
Or protein-protein interactions.
Yeah, well, I think quantum mechanics is most important for the sorts of things we've talked about today, is for catalysis, where you're making and breaking bonds, because it's, you can't really describe that sort of process using purely classical physics model. And so we've worked quite extensively with Ken Hauk on sort of doing these kind of hybrid quantum. Ken's group has been doing these hybrid quantum chemistry calculations to try to understand what's going on inside our designed enzymes. And to actually come up with these ideal enzyme active site configurations we can use in design that we call theozymes.
So there are still about 20 questions left, but we've gotten through 25. I think we just need to be respectful of time so we don't end up running out of it. So if it's okay with you, I think maybe you can answer a few more by typing, and I can get started.
Yeah, no, that's great, Jeff. Why don't you get started? And people can also feel free to email me. And thanks, those are great questions.
And by the way, thank you again, David. That was incredible. Hard act to follow for sure.
Looks good.
So I thought what I'd do today is to talk about some pretty fundamental work that we've done in both understanding proteins and understanding cellular protein homeostasis that, when extended, can be used to make pharmaceuticals. So since the inception of my lab, we've been very interested in neurodegenerative diseases that lead to dementia, which of course is defined as the impaired ability to remember and think that's so serious that it impairs the quality of daily life. So the diseases that we've been most interested in are the top three. The disease that affects the most patients and their caregivers, of course, is Alzheimer's disease, followed by Parkinson's and the transthyretin amyloidoses. And I'm going to talk a little bit about the transthyretin amyloid diseases first, and then Alzheimer's disease. So when we started working on the transthyretin amyloid diseases, the thinking was that amyloid infiltration of the extracellular space of postmitotic tissues caused their degeneration. And I think that's partially true, and we'll get back to that in a little bit. One of the things about the transthyretin amyloid diseases that we now think is pretty general is that these diseases affect organ systems like the heart, the peripheral and autonomic nervous systems, and the eye first. And then with time, they compromise the central nervous system. And of course, the common pathological feature of all these neurodegenerative diseases are amyloid fibrils, which are defined as cross beta sheet structures where the beta strands line up in a parallel mode. And through the work of Rob Tico and David Eisenberg and others, we now know that the side chains are very intimately packed in these amyloid structures as well. So transthyretin circulates as three forms in vivo. A very small proportion carries thyroid hormone. About half of the protein is unliganded, and roughly the other half carries haloretinol binding protein. And it's the tetrameric form of transthyretin that doesn't bind any ligands that has the possibility to aggregate in the circulatory system and in the central nervous system. In other words, it's the ligandless less form of this protein that's at risk for aggregation. So when we started this work, we wanted to investigate the extent to which the cellular biology played into these diseases. And that was rather unsatisfying in the sense that most cells secrete mutant forms of transthyretin roughly equivalently to the wild type protein. So that didn't seem to be a significant factor. But there was a lot of evidence that aggregation might be critical to the transthyretin amyloid diseases. And so we started to investigate the mechanism by which transthyretin aggregates. And perhaps the most important thing we found was that dissociation of the tetramer is actually rate limiting for conformational changes in the monomer that allow this protein to make lots of different aggregate structures. And what we found pretty early is that transthyretin, unlike a beta or alpha synuclein, forms myriad of aggregate structures. And amyloid fibrils can even be not the most prominent of the aggregate structures. And so it brought up the issue in terms of understanding structure proteotoxicity relationships that we really need to start to think about these other non-native structures that are formed. So when Ted Foss was a graduate student in my lab, he did a lot of beautiful protein engineering to try to understand how it is that the transthyretin tetramer comes apart. And it turns out that it comes apart about the dimer-dimer interface that I'm tracing out with the laser pointer. And this was a fortunate discovery in that this is the two sites where small molecule ligands can bind. And Ted revealed that this is the weak link in transthyretin aggregation. And so in principle, it became apparent almost immediately that we could fashion ligands to bind to these unoccupied sites to further stabilize the tetramer against dissociation, which was rate limiting for aggregation. So the idea simply is that you take advantage of these unoccupied binding sites, you make high affinity ligands, and we reason that this was a conservative approach in that clinical success didn't presuppose which aggregate structure was driving neurodegeneration in humans. So as many of you know, a lot of presupposition has happened in the Alzheimer's field. The notion that amyloid fibrils are the main players in driving dementia and other types of cognitive decline. And many companies have already spent tens of billions of dollars trying to clear amyloid fibrils, and they do it successfully, yet the patients don't get better. In contrast, the stabilizer approach blocks the entire process of aggregation, but only from newly synthesized protein. So the risk of this strategy was that, well, if the aggregates that are already there are irreversible or intractable, then the patients might not benefit so much. So I think one of the main questions that still kind of haunts many people in this field is, okay, so why does protein misfolding lead to neurodegenerative diseases, right? What's the fundamental mechanism of neurodegeneration? And I think David beautifully showed us that protein conformations enabled their function. So this is an example of a GPCR that binds to a certain conformation that the GPCR can adopt. Once the small molecule stabilizes that conformation, another protein can bind and signaling can occur. So the idea is that if you have proteins that can adopt myriad of unnatural conformations, then myriad unnatural functions result. And this might not be a problem if it happens for a week or a month, but over the course of 30 years, this is most likely what causes these diseases. And there's lots of evidence for, immune system hyperactivation, consumption of proteostasis capacity, mitochondrial dysfunction. People don't like these kind of mechanisms, right? That is multiple defects combined leading to a major pathology. But I'm afraid that's what we're likely dealing with here, and we have to learn to live with. Okay, so getting back to transthyretin. So now it remains to make high affinity ligands for the protein. And when Evan Powers was a postdoctoral fellow, he rationalized that, hey, structure-based drug design was actually envisioned and demonstrated using the protein transthyretin by Colin Blake, Jeff Blaney, and colleagues at Oxford. They had already published a lot of ligands that were thyroid hormone agonist or antagonist. So we started with those learnings to make small molecules that were not thyroid agonist and antagonist. And we ended up making about 1,000 molecules to ultimately come up with a drug tafamidis. Probably solved upwards of 150 crystal structures. While we were busy at work coming up with the lead candidate for patients, Teresa Calejo made a really important observation in her patient population in Portugal. So she had been taking care of thousands of patients who harbor this VAL30 mutation on one allele, which leads to an early onset peripheral and autonomic neuropathy, and later leads to dementia. And she recognized that there were a subset of her patients that didn't develop the disease, whereas some siblings did. And she recognized that there might be another mutation involved. And she sequenced these individuals, and sure enough, they had a mutation on both alleles. They had the V30M pathological mutation on allele 1, and they had a 3-119 to methionine mutation on allele 2. And we hypothesized that this incorporation of these 119 met subunits might super stabilize the tetramer thermodynamically, and that's how they protect against the transthyretin amyloid diseases. But in fact, that's not true. So when Per Hammerstrom was a postdoctoral fellow in the lab, he showed that the thermodynamic stability of the 119 met tetramer wasn't that different from wild type. But what really differed was the kinetics of dissociation. The idea being that incorporation of these 119 met subunits into tetramers otherwise composed of disease-associated subunits destabilized the transition state, effectively locking the protein in its tetrameric form where it couldn't get into trouble by dissociating and aggregating and causing tissue degeneration. And in fact, he showed both by looking at tetramer dissociation kinetics and fibroformation, which is shown here, the more protective subunits that you incorporate, the slower the process of tetramer dissociation is. So this was a really nice demonstration that this was predominantly a kinetic effect. Now, as chemists, of course, we don't know very well how to engineer transition states, although clearly David is getting much better at that. But it wasn't too hard to envision how we could target small molecules that selectively bind to the tetrameric native state and not to the dissociative transition state. And therefore, you can tune the energy barrier of the transthyretin tetramer through small molecule ligand binding. So this is a special type of kinetic stabilization called kinetic stabilization that David Baker actually studied for his PhD thesis, albeit in his case, an amino acid-based origin. Here, it's a small molecule-based origin where you make the activation barrier very high for a process that you don't want to happen. So we measure kinetics in human fluids by looking at subunit exchange between tetramers. So the endogenous tetramer from patients here is shown in black. And then we add a recombinant transthyretin tetramer that have these dual flag tag extensions. And this allows us, using chromatography, to discern whether the tetramer we add and the tetramers that's present exchange subunits in the presence of the kinetic stabilizing small molecules. Now, this would never work chromatographically if we didn't have a small molecule to render the transthyretin tetramers fluorescent. And Sung-Wook Choi discovered this molecule a few years ago, which upon reaction with transthyretin renders the conjugate fluorescent. So you can do chromatography in biological fluids that have 4,000 or 5,000 proteins. So this is actually a Nobel laureate who lived almost 15 years with cardiomyopathy that was on tafamidis. And he was incredibly gracious and allowed us to do some critical experiments for dose setting in humans. And the idea is that as you increase the dose, you slow the tetramer dissociation rate. And we realized that 60 milligram oral dose of tafamidis almost stopped the dissociation of transthyretin, as is shown here in these subunit exchange-based experiments using the methodology I outlined in the prior slide. Okay, so then it was time to jump to humans because there were no animal models. And we argued to the FDA that Teresa Coelho's data already showed us that in humans, if you stabilize the protein, you prevent the disease. So it was rational to think that we could at least slow the progression by treating with a small molecule that phenocopied the interallelic trend suppressor mutations. So we decided on a 20 milligram dose, which we didn't really like very much, but the FDA loved because they were worried about us creating a lysosomal storage disease, which turns out to be baseless, as we learned through the years when we take the dose up. But in any case, you know, as in most clinical trials, the tricky part is to pick the endpoint. And here we picked the neurologic impairment score, which gave me a lot of sleepless nights because every idiopathic polyneuropathy trial using this score has failed. But we rationalized that these patients progress faster, and that turned out to be true. So the drug-treated group for the first 18 months is shown in the filled circles, and the placebo group is the open squares. Even in 18 months, we got a nice separation. And then as trials usually go, the placebo group rolls onto drug, and their slope was decreased. Now, what's more important is now we can look back, since most patients now have been on drug a decade that were part of the trial. And if you look at death versus time, you can see that tafamidis is really superior to liver transplantation, which is a form of gene therapy where you eliminate the mutant protein, and I'll have more to say about that in a minute, versus not being treated, which most of the individuals die within 10 to 15 years. So not only is the quality of life increased, but also lifespan is clearly increased with this drug. Now, if you develop cardiomyopathy from the wild type protein, that disease progresses very fast. And these were fairly late stage individuals that we recruited for the clinical trial. And you can see an important take home here, which is that if you look at demographically identical groups, and the placebo group is on placebo for 18 months before they roll on to tafamidis, whereas the tafamidis group is on drug from T equals 0. You can see that treating the patients early is absolutely critical in these diseases. And I suspect that ultimately explains many of the failures in Alzheimer's and Parkinson's, because those diseases are harder to get people on drug early, and we can talk about the reasons for that in the discussion section if you'd like. So treating early is critical for being successful in these disorders. And again, this is a Kaplan-Meier curve showing that early treatment is superior to even delaying by 18 months. I don't want to take the time to go through the 6-minute walk test and the quality of life, but the bottom line is that treatment with tafamidis improves significantly both of these important parameters that the FDA insisted on. Now, it turns out that we have an embarrassing number of drugs that modify transthyretin amyloidosis now. So 6 years after tafamidis was approved, both Alnylam and Ionis got agents that degrade the messenger RNA of transthyretin approved. And these drugs, like tafamidis, are quite effective for treating the transthyretin amyloid diseases. As I mentioned, when we treat only the peripheral symptoms, as we do with the messenger RNA lowering agents, or as we do with liver transplantation, where we replace the mutant transthyretin with wild type by swapping out a liver encoding wild type TTR, then the peripheral symptoms that historically have killed these patients are slowed down or stopped dramatically. But the choroid plexus also secretes transthyretin, and those treatments don't affect the brain pathology. And the brain pathology is slow onset, almost certainly because the transthyretin concentration there is only 200 nanomolar relative to the 4 micromolar concentration found in blood. And remember that aggregation is concentration dependent. And so this order of magnitude difference in concentration is undoubtedly a contributor to the tenfold delay in the central nervous system diseases. And this is almost identical what's seen in Parkinson's disease, wherein the periphery is affected a decade before the central nervous system is. So nobody, I think, by and large, was expecting people treated by liver transplantation to go on and develop dementia, because except for the rare familial cases that presented with dementia, it wasn't generally perceived that this was going to affect most of the transthyretin amyloidosis patients, but it does. And Yoshi Sikajima showed by amyloid imaging in the brain that about a decade after you do the transplant to slow down the peripheral progression, you end up with brain disease, which manifest as focal neurologic episodes. These are really bad. So if you're driving your car, all of a sudden you just lose the ability to think for a few seconds to a few minutes. It's like you lose consciousness. And so this really affects people's quality of life, obviously. Moreover, these individuals are demented. So even early on, 40% of them have a dramatically lower Minty-Mendel exam score, which is used in Alzheimer's disease. Now, fortunately, and we certainly didn't design this, okay, tafamidis has a decent brain uptake even at the 20 milligram dose. So the concentration of the drug is about a half to 3/4 of what the protein concentration is. And since the dissociation constant is 2 nanomolar, that means you get pretty much almost full occupancy relative to the drug concentration in the brain. And so far, people who've been on tafamidis for more than 12 years have not developed dementia. And we certainly hope that this holds. And I should say that for cardiomyopathy, now they're using an 80 milligram dose rather than a 20 milligram dose, because as you saw in our Nobel laureate friend, this stabilized transthyretin completely. So we're hoping that we probably won't get a linear increase, because that isn't how pharmacokinetics work, but maybe we'll get 3/4 of the gain we expect, and that should be really profound. And this is really important because the million patients who have transthyretin amyloidosis mostly are wild type. The disease starts with a cardiomyopathy, and if patients are fortunate enough to survive that, they end up with cerebrovascular bleeds and dementia. So we're hoping that tafamidis at the higher dose can prevent both of those, but it's too early to tell. So what have we learned through all this, right? I think, firstly, we've learned that dissociation and aggregation of newly synthesized transthyretin is the main driver of pathology, at least with regard to death and serious disease that puts people in the hospital. Now, remember, these patients were symptomatic when we started treating them. And in fact, there's no measurable change in the amyloid load in these patients. So I'm not arguing that amyloid is a good thing. It is clearly not, especially in the heart, when it stiffens the heart and makes the heart less operable. But it's probably not what's causing people to have major dementia and major organ failure. Rather, it's the diffusible structures that we're really lousy at characterizing now that may, in fact, be the main driver of these diseases. So in summary, it's the process of aggregation from newly synthesized transthyretin that I would posit causes the postmitotic tissue degeneration in these disorders. Okay, so lots of companies now are making stabilizers, kinetic stabilizers for a myriad of proteins, but And we've done the same for light chain, and we'll be in the clinic in the next 24 months to test that drug. But we realize that there are almost 50 of these diseases, and it's a lot of work to make a stabilizer for one. So we wanted a more general solution. And for that, we decided to think about how we could pharmacologically modulate the protein homeostasis network in humans to accomplish this. So what's the protein homeostasis network? It's a compilation of about 2,500 proteins that help proteins fold, traffic, and degrades them when they need to be degraded. So as a polypeptide comes off the ribosome, we now appreciate that there's a kinetic competition between protein folding with unimolecular kinetics, protein misfolding with unimolecular kinetics, and concentration-dependent protein aggregation. And the proteostasis network through chaperones and the ubiquitin ligase system and the lysosomal autophagy pathway are very good generally at getting rid of misfolded proteins, aggregated proteins. It's not just David Baker's design proteins that fold with less than 100% efficiency. Most of our proteins fold with well less than 100% efficiency. So it's critical for this compilation of proteostasis network components to direct misfolded and aggregated forms to degradation. So we thought that the degradation pathways are really underappreciated in these neurodegenerative diseases. So in particular, autophagy has not been tested yet in terms of its activation as a way to ameliorate these diseases, but autophagy is fantastic at getting rid of big protein aggregates, protein RNA aggregates, and so on, because the lysosome comprises 65 hydrolase enzymes that degrades all of the macromolecules we know of, right? Nucleic acids, carbohydrates, lipids, and proteins. And all of those things comprise protein aggregates. So we thought that might be important. So the autophagy process is rather complicated, but I'm going to describe it at a high level today because I think that's sufficient. If you want to know more, of course, we can discuss that. So autophagy starts with the formation of a phagophore, which is a double membrane structure that's studded with receptors that bind to things like proteins, organelles, and large macromolecular machines like the ribosome. Then ultimately, this double membrane structure is expanded, and in 3 dimensions, it encompasses the substrates to be degraded. And then the autophagosome fuses with the lysosome such that the contents and the membrane of the autophagosome can be degraded. So there's a lot of protein biochemistry and lipid biology that's underway here. Now, we got interested in this pathway because of the compelling human genetics. So there are mutations in humans that compromise the efficiency of every step of the autophagy pathway. And these mutations, by and large, don't cause a phenotype in young people. They only cause a phenotype in older individuals, and that phenotype is almost always the same. It's neurodegeneration. So we hypothesized that since impaired lysosomal flux causes neurodegeneration, that maybe if we could enhance lysosomal flux in the aged, that would be a way to generally treat neurodegenerative diseases associated with aggregation of proteins, nucleic acids, and lipids. The problem was no one had ever really done a high throughput screen for discovering autophagy activators that looked at 1 million small molecules. People had figured out ways to look at most a couple 100,000, and that took a long time, given the way that they did it. So there's a beautiful paper in Nature from a group at Harvard showing that lipid droplets are cleared by autophagy in most cells. And you can make lipid droplets prominent simply by feeding cells a fatty acid. In this case, we used oleic acid. So we fed cells with, we fed a certain glioblastoma cell oleic acid for 24 hours. And then we screened for compounds that gave the phenotype on the bottom left, that is very few lipid droplets. And fortunately, lipid droplets can be easily visualized in cells using fluorogenic ligands like BODIPI. Okay, so by screening candidate small molecules at 5 micromolar in a high content imaging screen, we are able to go through 1 million small molecules looking for the phenotype on the right versus the vehicle or say a autophagy inhibitor phenotype on the bottom left. And we ended up with about 200 small molecules after a whole bunch of counter screens where we threw out compounds that were hits because of fluorescent artifacts and the like. And fortunately, most of these compounds cleared lipid droplets really well. But there are other ways, as Chuck knows, to clear lipid droplets, right? There are lipase enzymes that do that. And so, in fact, not all of these compounds that we discovered are autophagy activators. Some of them are incredibly good at activating cytosolic lipase enzymes for mechanisms we're trying to sort out. But for those molecules that work by enhancing macroautophagy, they should recruit this critical protein LC3 from the cytosol to the phagophore. And in so doing, LC3 is proteolyzed and lipidated. And so its molecular weight goes down on a gel, that is, it runs farther, in part because a few amino acids are cut off and in part because of the lipid being present. And so you can see nice activation of autophagy through LC3 lipidation. But as you all know, running gels is painful and you don't want to do that for 200 small molecules. And so Ivan Dickick at Alexandra Stolz in Germany automated this beautiful assay that came out of Japan, whereby you utilize an effector protein for autophagy, which is a protease, to cleave GFP LC3, which goes on to perform its effector role in autophagy. And that is cleaved away from RFP LC3, which is minus a glycine at the end. And that glycine's critical for attaching phosphatidyl ethanolamine, which is the lipid that sequesters it to the membrane. So the red protein stays in the cytosol and the green protein gets degraded if you've activated autophagy. And so you can look at that ratio, which we've done for all of our compounds over 72 hours to see which compounds activate autophagy. And these really broad red streaks are what we want. So these are compounds that even work at 50 nanomolar in cells to activate autophagy. And we've been lucky enough to find a few of those. Now, importantly, there are really good activators of autophagy, namely rapamycin and its homologs that work through inhibiting mTOR. So mTOR is a kinase that goes on to phosphorylate a lot of substrates, and including those that block autophagy. And so if you inhibit the mTOR kinase, you remove the negative regulation of autophagy and you activate it. The problem is that, as many of you know, rapamycin is a drug that inhibits the immune system and it's used to ameliorate organ rejection. And so the last thing you want to do in the elderly, I think, is to treat them with an agent that impairs their ability to fight infectious diseases, since they're already at higher risk for these diseases. If you can. Now, that's not to say that we can't learn a lot about autophagy from rapamycin. And in fact, that's ongoing. So, one of the high bars that needs to be met in this field is to actually look at lysosomal kinetics. And Steve Finkbeiner and his colleagues figured out a beautiful way to do this by fusing this LC3 effector protein to a GFP that's photoswitchable. So you can do a pulse chase experiment with and without drug. So, the mean half-life of LC3 is about, 34 hours in the absence of drug, just vehicle being present. And you can see that rapamycin doubles or more the rate of lysosomal flux, and molecules like A5 nicely show a dose-dependent decrease in lysosomal flux. And this is exactly the kind of behavior that we want to see. And notably, again, this compound is active at 50 nanomolar in human cells and human neurons. That was a primary mouse neuron. So with Steve Haggerty's lab at Harvard, we decided to test this particular molecule, A5, in P301 neurons. And this mutation is important in tau pathology, which is the major driver of the dementia in Alzheimer's disease after a beta aggregation initiates tau aggregation. And so the nice thing we were able to show is that A5, through autophagy activation, nicely depletes both soluble tau and insoluble tau. And notably, A5 doesn't deplete the majority of the proteome based on really careful proteomics experiments, but it's really good at depleting tau. And the Haggerty lab has already showed this with their rapamycin analogs. And critically important, this molecule also gets rid of aggregated tau. And of course, that is critical because it's thought that these phosphorylated forms are actually the drivers in dementia in Alzheimer's disease. Moreover, molecule A5 can spare neurons from neurodegeneration and death caused by additional stressors like rotenone, which is a mitochondrial decoupler, NMDA, which is a hyperexcitability inducer in the brain, rotenone, which compromises the mitochondria. Well, yeah, you already saw that. So, so the bottom line is that, you know, in the absence of autophagy activation by A5, you get about 60% neuronal loss, but in the presence of A5, you can preserve those neurons. Now, these compounds are hits, so they're a little toxic at the high concentrations, and that's why we see that dip there. And that's common when you use more than one drug agent in combination, that the toxicities are exacerbated. Now, one of the other features that I only want to present in the last couple slides that are common in neurodegenerative diseases are these axonal inclusions. Now, Larry Goldstein's lab at UCSD was the first to show these and their mechanistic basis, and that they involve, their formation involved macromolecular motors and the like. Now, importantly, you can see in this movie that in a neuron where the normal prion protein is labeled, you see nice retrograde and anterograde trafficking of vesicles along the axon. That's what's supposed to happen, right? Things that are needed at the synapse race towards the synapse, and things that need to be shuttled back to the cell body do so. In the case of expressing a prion protein with an expanded polyproline repeat that induces the prion protein to aggregate, you can see that trafficking is really slowed down and it's really compromised. And Sandra Ancalada's lab showed in a beautiful paper that slowing down trafficking like this causes neuronal death, not surprisingly, because of the need for critical factors that are made in the soma at the synapse. And what was really exciting to us in these primary neurons, using turf microscopy imaging, we were able to show that drug treatment reduces the amount of these axonal endosome delimited aggregates back down to baseline levels, and that this happened at concentrations as low as 50 nanomolar, and that it was dose dependent. And what Sandra's lab showed that's even more remarkable, I think, and represents a fundamental discovery, is that A5 induces autophagy as discerned from these dyes that light up acidic lysosomes by drug treatment. So in the absence of drug treatment in these vesicles that have a lot of aggregates, you don't see any acidic lysosomes present. But in the presence of the drug, you see a dramatic increase in acidification and functions of the lysosome. This is notable for two reasons. First, it was arguable whether there were even lysosomes in axons. Other people had argued strongly against that. People had thought that maybe everything has to go back to the cell body to get degraded in the lysosome, but that's clearly not true. Okay, let me close there and say that, I've showed you some protein chemistry that led to an FDA-approved drug, and I showed you how by understanding how cells manage protein homeostasis, that maybe we can someday get to a drug that enhances lysosomal clearance of not only proteins, but the lipid accumulation and other things that are characteristic of neurodegenerative diseases. And most importantly, thank you very much for joining us today and listening to David's wonderful talk. And I hope I was able to get somewhere close.
Yay, great talk.
Well, thank you, Jeff, for an amazing and super impactful talk. I guess while questions are coming up, I had a couple. One is, what do you think the prospect understanding the process? I guess the last example you gave with the interference in axon trafficking is an example of sort of identifying a specific process that is biological process that is going wrong, sort of a molecular mechanism. What do you think the prospects are more generally for understanding where the toxicity is coming from? And the second was just a very specific question. Do you know what the molecular target for A5 is?
Yes, I think we do know the molecular target for A5. I mean, this is literally unfolded in the last week. And so I'm a little reluctant to announce what we think it is, but this has been a 3-year exercise. I mean, we knew we had to have a pretty good targets because of the potency of this compound. And in fact, the target has been linked to autophagy in the past, and we just need to link it back with genetics so that I can become more confident that we really got it. I mean, with regard to your first question, I think that's one of the central issues in this field, right? And that we need to begin to understand how it is that the process of protein aggregation and lipid accumulation and aberrant phase separations are causing the demise of neurons. Part of the problem, as you know, is that we don't have good probes for aggregate structures that are not amyloid-like. And I'm hoping that cryo-electron microscopy will start to reveal some of those structures. And then using the beautiful tools that you've outlined today, we can make protein-based probes for these various structures and start to correlate what drugs do with what's happening in patients. And that's probably the only way that we're ultimately going to get there. And then, then there's the really fundamental studies that neurobiologists are doing. Like I think, what Sandra's work in the axons have shown is that, yeah, if you build up these endosome delimited aggregates of the prion proteins, the neurons die. And if you can get rid of them, trafficking is normalized and the neurons don't die. Now, whether that's going to predict whether we can treat a prion patient, only a clinical trial will tell. But I think, I believe in basic science, as I know you do, and Chuck. And so, having that understanding gives us a good shot on goal in the clinic.
Great. Yeah, certainly. It's got to be one of the most important areas in all of protein science. So, yeah, so keep up with the great work. Let's see, we have a couple questions. An anonymous attendee asks, the AL inhibitor you are developing is universal for kappas and lambdas, question mark. Does it stabilize the Bence-Jones type of dimer?
Yeah, it's a very good question. So the challenge with light chain amyloidosis is each patient has a different light chain sequence. These light chains are predominantly lambda light chains, but there are other forms as well. These light chains are formed in all of us, these light chain dimers, in excess with antibody secretion, but those light chains just get cleared and there's no problem. In light chain amyloidosis patients, those cells start to proliferate. So there's a cancer component, which generates a lot of light chain because there's a lot of those clonal cells. And moreover, the sequence of those antibodies is aggregation prone. And the process of aggregation causes the demise of various organs, especially the heart. And that leads to patient deaths prematurely. So we've been able to very fortunately find a small molecule binding site that wasn't previously known in light chains, although David Eisenberg got us really close to that site through some beautiful work he did. And we've been able to find small molecules that use side chains that are present in almost all antibodies. And so we have a general solution. And so it's a kinetic stabilizer like tafamidis, that is, it binds to the native tetramer and not the misfolding transition state very well. And so hopefully it will work in patients. And, you know, with a little luck, we'll be in patients in 18 months.
Great, thanks. Let's see. So Arun Agrawal says, very interesting work. In transthyretin trials data, you indicated that treatment should be at early stage. How do you identify and what symptoms would be used to identify early stage?
Yeah, this is the huge challenge in all amyloid diseases. It's easier when you have a family history and you know that the grandmother and the parents develop the disease. So neurologists follow these patients yearly in careful neurological exams. We're starting to come up with better MRI and PET-based imaging methods to screen people at risk. But the problem is that most amyloidosis patients have no family history of high risk. And so generally, it's still not possible to diagnose them early. We need a blood test like the cholesterol prediction test, and we don't have them right now.
Let's see.
I have a question.
Go ahead. Yeah.
So in both projects where you did screens to find hits, usually there's a step that involves considerable medicinal chemistry. Did you do that work in your own lab, or how did that work?
Yeah, so we've done a lot of medicinal chemistry over the years. Yes, so the answer is we do enough medicinal chemistry to get to a lead candidate. And then at that point, we often form a company and license it out to a professional organization to do the drug development. There's just so many steps that the FDA doesn't believe that academia can do well. So it's almost pointless to try to do it. Even if you could do it, they'd make you do it over.
Thank you.
See, Shukbong Zhang asks, can people discover specific monoclonal antibodies to treat amyloid disease, neurodegenerative diseases?
There are several of those in clinical trials right now, including monoclonal antibodies that stabilize protein native states. And my sense is that those should work in the clinic based on, analogous mechanisms that have read out positively in clinical trials. The antibodies that recognize non-native structures and bring macrophages or microglial cells to destroy them have so far had a checkered past, in the sense that mechanistically they accomplish what was desired, but the clinical outcomes haven't slowed or stopped dementia.
Now, just let me ask a question. Do you think that's because they've come in too late, or because they aren't decreasing the concentrations of the newly synthesized forms that are toxic?
Yeah, that's a question that nobody can answer right now, I think. And it's a critical one. And I really think the problem in Alzheimer's and Parkinson's, by and large, is that we're treating them way too late. I mean, there's evidence emerging from several labs, including David Holtzman's lab and Alison Goet's lab and others, that hyperactivation of the immune system in the later stages of Alzheimer's and Parkinson's is a substantial driver of the pathology. So if you just fix the proteinopathy, that's probably not sufficient, because now the immune system is destroying neurons by virtue of overactivation. We even see this in early stage patients, David, which is scary, right? So both the Alnylam Ionis drug and the Pfizer drug that we discovered, tafamidis, those drugs only treat 70% of early stage transthyretin amyloidosis patients. The other 30% are untreatable. And so even when you can get drug on board earlier, you still lose a subset of patients. And we're trying to figure out if that's due to hyperactivation of the immune system or what. We don't know yet.
Do you think there's potential for dampening the immune response in the late stage to mitigate symptoms, or is it just too systemic?
Yeah, I do think that's a very promising strategy, especially when combined with a drug that addresses the proteinopathy component.
Thanks. Anonymous attendee asks, is there any danger in decreasing the concentration of soluble tau?
Well, that's a fantastic question that a lot of people are addressing. And so far, microtubule dynamics have not been changed that much by drugs that lower tau levels pretty substantially. But I think there is a limit there. And people in industry who are trying to lower tau to treat tauopathies are very cognizant of that, trying to find the right balance, kind of the Goldilocks principle, right? You want to knock it down enough, but not too much.
Now, why would you showed that you could enhance the degradation of soluble tau as well as the amyloid form, I think. Yes. And why would the soluble form why would that be enhanced if it looks normal?
We really don't know. I mean, one possibility is that it's not normal owing to some post-translational modifications that direct it to a certain lysosomal receptor. Interestingly, that occurs to a lesser extent in non-familial iPSC-derived neurons. So I think there may be something to that hypothesis I said. And I don't think it relates to the mechanism of A5 activating macroautophagy, because Haggerty's rapamycin analogs do exactly the same thing. And what's even more surprising in terms of both drug candidates is that the tau levels stay down for like 2 weeks after one treatment, may even be longer. We just haven't looked longer. So there's some really interesting effects going on that we don't understand yet, which we're trying to collectively figure out.
Yeah, and let's see, we have time for one more question. Anonymous attendee asks, in the context of the prion model, do you believe that small molecules, antibodies, and/or nanobodies could be developed against pathological monomer seeds? How has this work progressed for TTR?
David, I'm sorry that my internet went a little blank there for a second. Can I impose on you to ask it again?
Sure. And that might have been on my end also. In the context of the prion model, do you believe that small molecules, antibodies, and/or nanobodies could be developed against pathological monomeric seeds? How has this work progressed for TTR?
Yeah, I do think they could be addressed. And, you know, David, you're working, I think, on something very similar where you take structures that ultimately lead to those monomeric seeds and pull them out through protein-protein interactions. So it's early days. That hasn't been demonstrated in the clinic yet, but I see no reason why that mechanism of action couldn't have utility in the clinic.
Right. Thanks, Jeff. Well, we're at 11 o'clock. I don't know whether we should stop here or keep going. Luca and Chuck.
Yeah, I think probably we should go ahead and wrap it up and just get just do it that way. But I mean, these are two fantastic talks and fantastic discussion. We're so grateful to you for